Assalamualaikum everyone. In today's video, we are going to discuss another important biochemistry practical, and this practical is about the detection of carbohydrates. In the previous lecture, we have discussed that how we detect carbohydrates by a famous test which is called as Mollis test. But in this test, we are going to detect our carbohydrates by another important test, which is iodine test. So in the previous Mollis test, we have discussed that it has ability to detect all the carbohydrates, whether it is aldoses, whether it is ketoses, whether it is monosaccharides, oligosaccharides, polysaccharides, or disaccharides. So it can detect all the carbohydrates. So the Mollis test is for the differentiation of a carbohydrate or a non-carbohydrates compounds. But in the today's video, which is about the iodine test, in this test we detect only polysaccharides. So it is only used to differentiate that whether it is polysaccharide is or not. So what is the principle? First we discuss the principle of this test. So basically, you know that polysaccharides are helically coiled upon each other. So due to this coiled nature, they have ability to absorb small molecules of iodine on its surface, and due to this absorbance they can form a complex of color. So at least eight monosaccharides united to form a linear chain to give the color complex. So this is the condition for this test, that we should have a polysaccharide which have minimum eight monosaccharide sugars in them, and maybe more than eight, but minimum eight is definite to form this color complex. So the reaction only occur in the acidic or neutral medium. This is the second condition, that we need the acidic condition or neutral medium. If you cannot give the basic medium to it, because whenever you give the basic condition, its color complex will not form. So heating of starch, you know, starch is a polysaccharides in presence of concentrated HCl causes its hydrolysis, and it convert into the glucose. So what will be the effect of acidic and neutral medium? It explained here. If alkali effect, any basic effect, disturb the secondary valencies of polysaccharides, thus the iodine is released from their surface and therefore color disappear. So if you take this whole process or iodine test in the basic medium, then what will occur? Actually, in the basic medium, the secondary valencies of polysaccharide disturb. Due to this disturbance, has no ability to absorb iodine molecules. So due to this non-absorbance, what happened? That the color complex will not form, iodine will not absorb, and no color, the color disappears. But if we have strong acidic condition, then the secondary valencies are not disturbed, and there is absorption of iodine. Due to this absorption, the color complex will be formed, and we get identification point that this solution contains monosaccharides, polysaccharides in them. So another important condition for this test is the heating effect. You should take very care of the heating of your solution, because heating can also disturb the color complex. So heating dissociate the iodine and polysaccharide complex, and the color disappears. So if you heat your solution, this color complex will be demolished, disappear. On cooling, reappear and form the color complex. Okay? On cooling, whenever the temperature decreases, the color complex again appear, and you will see the color PPT. But except dextrin, actually whenever you heat the dextrin, its color disappear, but on cooling it never restore its color back, because due to heating its structure changes. So therefore there is a exceptional case for the dextrin, that it will not recover back its color on cooling, but all the other polysaccharides recover their color back on the cooling. So you will take care of all of these conditions for this test. Now we move on to the next page, and we discuss how we prepare this iodine reagent. Because of heating changes the dextrin molecule and losing the property of absorption. I have explained to you that the dextrin change its molecular structure, and due to this change the color of dextrin will not restore whether it is cooling back. So now you have to prepare this iodine reagent. First you have to write the apparatus and the material you want to perform this reaction. So first you need the iodine reagent, and I will show how you prepare this. Then you should have some solutions, for example glucose solution, lactose solutions. These solutions could be monosaccharides, could be disaccharides, and could be polysaccharides. Actually we want to differentiate the disaccharides or oligosaccharides from the polysaccharides. So you will take the glucose solutions, lactose, glycogen, and unknown solution, and 2% carbohydrate solutions of every 2% ग्लूकोज सोल्यूशन मतलब ये सुक्रोज स्टार्च एंड डेक्सट्रिन ठीक है. So now how you prepare this iodine reagent. So first of all you will add few crystal of iodines ठीक है to the 2% potassium iodide solution. Mean first of all you will take 2% of iodine potassium iodide. How you prepare 2% solution of it? You will take 2 grams of potassium iodide and dissolve in 100 ml of water. You will get the 2% solution. Now in this 2% solution you will add the crystals of iodine in it, and your iodine reagent is prepared. So simple. So now you have to perform the experiment and you have to write up the procedure. Now what you do, you take this iodine reagent and add all small drops of iodine reagent in all of these solutions. So first of all, add a few drops of iodine solution or reagent, about 1 ml of the test tube solution. So you will take 1 ml of starch solution, 1 ml of glucose solution, 1 ml of lactose, glycogen or other solutions. Now when you add in the 1 ml starch solution, you will going to add few drops, 2 to 3 drops of iodine solution. So what will happen? The starch will give the blue color. You know that starch is a polysaccharide, so therefore the color appear and the blue color appear. So the blue color appearance is indicating that this is a starch. No other polysaccharide give this blue color. Only starch give blue color. So therefore you can identify which of the polysaccharide it is. So blue color always going to detect that it is a starch. So iodine is entrapped inside the coils and the complex give intense blue color. Now the other solutions which we have taken give also some colors. For example, dextrin is also a polysaccharide. It is a polysaccharide of glucose and contain a lot of glucose molecules in it. So it has a red color in it. Glycogen has purple color, while archaeodextrin, maltose, glucose, you know very well that these are monosaccharides. So therefore they are giving no color, indicating that this solution is not a polysaccharide. So in that way we differentiate that whether it is a polysaccharide or not, and the specific colors of the polysaccharide indicating that this type of polysaccharide. So now you define some effects of pH and temperature on this test. First you have to add base in that solution, then you will add some acid in that solution, and you note the effect of pH. So you are actually disturbing the pH, and you are going to note that whether it is affecting or not. So when you add, first of all you are going to add the base, and 10% solution of NaOH is a base to the iodine test, और drop by drop डालेंगे and the color disappear. Now you add concentrated H2SO4, which is an acid, drop by drop till the color reappears. So this reappearance and disappearance indicating that it is a polysaccharide. I explained to you, whenever you are going to add the base in that solution, it is going to disturb the secondary valencies, and due to the disturbance of secondary valencies, what happened, your sugar will not absorb iodine molecule, and due to the non absorbance of iodine there is no color. So whenever you are going to add acid, the secondary valencies does not disturb, and the iodine molecule easily absorbed by the helically coiled polysaccharides, and due to this absorbance, the colored form. So therefore, whenever you add base, colors disappear. Whenever you add acid, colors reappear. So the second effect is the effect of temperature. I have explained to you, whenever you heat, the colors disappear because heat changes the structure of molecules and enable the molecule or sugar to absorb the iodine molecule. So therefore, whenever you heat the test tube, iodine disappear. Now, if you cool the test tube, the colors reappear. But the exceptional case for the, yes, dextrin solution, because dextrin is not going to restore its color on heating, or sorry, cooling, because its structure whole disturbed. So therefore, it not going to restore its color. So the color of dextrin is red color. So it is not going to restore its red color after cooling. So the precautions of this test is that you should not take this test to the alkali medium, because in the alkali medium all the things will be going to disturb. Also you take care of temperature, because the negative at the higher temperature, the this test is going to disturb. So it is better to perform it or at the near the room temperature. Do not rise or lose the temperature, because temperature has effect on that test. So the last precaution is that iodine should not be added in excess. Whenever you add in excess, the rings which appear or the colors which appear disturbs and mix with themselves, and there is a possibility that this ring disappears, or a complex and other color will be formed due to excess of iodine reagent. So therefore you will be take care of everything, whether it is pH, whether it is the temperature, whether it is the access, and the limit you always remain in the limit for this test. So now I will explain the whole diagram and whole the procedure and how these colors appears. So next में हम लोग ये पढ़ेंगे. This is the representative diagram of this experiment, and this diagram will going to explain all the procedure to you. So in this diagram you will see this figure. It contains 6 test tube, and in the 6 test tube there is a solution, and every solution is of 1 ml. So first solution is of starch solution, second is of a glucose solution, third is glycogen, maltose, dextrin, and archodextrin solution. You have maltose solution, dextrin solution, and archodextrin solutions. Now you are going to add a few drops of iodine reagent. For example, I have dropped some iodine reagent in it, in this, in this, in this, in this, in this. So now you will shake this test tube, and after some time the starch will give blue color, glucose give no color, it remains same. Glycogen is going to give you purple color, maltose, because it is a disaccharide, so it will give no color, dextrin give red color to you, and archodextrin give no color to you. So in that way you will indicate this and this, it is polysaccharide, whether it this and this test tube contain the solution which are not polysaccharide. So in that way you are going to differentiate them. So always blue color indicate starch, purple color indicate glycogen, dextrin indicate red color. So in this way we have performed this experiment, and you will write that the appearance of blue color indicate that it is a starch solution. The appearance of no color indicate that it is a not polysaccharide. So in every test tube you will write the reason for the appearance of color and the reason for the no appearance of color. So this is the observation table for the preparation of iodine reagent, and you will write the name of reagent and the quantity. So you know the iodine reagent consists of two major agents. First is the iodine crystals, and second is the potassium iodide. So few iodine crystals and 2% solution of potassium iodide will combine up and form the iodine reagent. So this is the observation table number 2. In this observation you will write all the test solutions, which is glucose, starch, archodextrin, maltose, glycogen, and dextrin. You will write up the quantity, for example 1 ml of each solution, few drops of iodine reagent is added in the all the solutions. At the last you will write the result of color, whether it is appear or not appear. So in the last you will write up the result that iodine test was performed by different carbohydrates such as starch, glycogen, dextrin, and give a blue, purple and red color. Starch give blue color, glycogen give yes purple color, dextrin give red color, while the glucose, sucrose and lactose give no color. So in that way I hope कि आप लोगों को ये सारा लेक्चर समझ में आ गया होगा. If you have some confusions or some questions, you will write up in the comment section. I will answer to you, and please like my videos and subscribe my channel. And I hope you will subscribe my channel and don't cheat. You are not going to only सो आई होप कि आपने चैनल को सब्सक्राइब कर दिया होगा, वीडियो को लाइक कर दिया होगा. चैनल को वो ज़्यादा से ज़्यादा सब्सक्राइब करें ताकि मैं आपके लिए और इस तरह की वीडियोस लाती रहूँ और अच्छा से अच्छा कंटेंट लेके आती रहूँ. सो आई होप कि आप लोगों को लेक्चर अच्छा लगा होगा, और इसे अपने फ्रेंड्स के साथ भी शेयर करें, इसे अपनी फैमिली के साथ भी शेयर करें. आई होप कि आप लोगों को समझ आ गई होगी सारे लेक्चर की. फिर भी अगर आपको कोई डाउट है तो आप कभी भी कमेंट सेक्शन में कर सकते हैं, मैं आपको उसको ज़रूर जवाब दूँगी. और अगर आपको वीडियो अच्छी लगी है तो बेशक सब्सक्राइब ना करें, बेशक लाइक ना करें, बट कमेंट सेक्शन में अप्रिशिएट कर दिया करें. या अगर आपको कोई मिस्टेक नज़र आती है तो आप वो भी कहा करें ताकि मैं उसे ठीक करूँ और आपके लिए अच्छा सा अच्छा कंटेंट लेके आऊँ. सो आई होप कि सही हुआ होगा, वीडियो आपको पसंद आई हुई होगी. और टेक केयर ऑफ़ योर सेल्फ. मिलेंगे अगले लेक्चर में एक अगले एक्सपेरिमेंट के साथ. तब तक के लिए अल्लाह हाफिज़.